Hello everyone and welcome to this lectures on disease inheritance evolution. We are going to be progressing following the program as shown on the screen. We'll start with a brief introduction. Later we are going to look at the different types of disease inheritance which have been divided into two, the Mendelian disease inheritance and the non-Mendelian disease inheritance. Then we'll move on to evolution and then finally to speciation. Now, we are doing biochemical genetics. What is genetics in itself? So, genetics is actually the study of genes. What is therefore biochemical genetics? Biochemical genetics is the study of the influence of genes in certain metabolic pathways in the system. So, genetics is divided into three main parts. We have classical genetics that actually deals with inheritance. So, when we are doing Mendelian crosses, we are actually doing classical genetics. Then we have molecular genetics that actually deals with the structures, the multiplication and the properties of nucleic acids. Okay, and then we have population genetics that deals with evolution, speciation, and ecosystems. So in this course, we are going to do part of clinical or classical genetics. We are going to do part of population genetics. So under classical genetics, we are going to be looking at disease inheritance. While under population genetics, we are going to be looking at evolution and speciation. So there are some basic terms that we have to define which are commonly used in biochemical genetics, okay, so that we can better understand the cause. We have a gene. So a gene is that section of the DNA that codes for a functional protein, okay? So if the protein is not functional, it is not a gene, okay? So a gene is that section of the DNA that codes for a functional protein. Then we have an allele. Alleles are alternative forms of a gene, okay? If we take Mendelian crosses, the experiments that he did with pea plants, okay, we have the allele for tallness, okay, and we have the allele for shortness, okay. So these alternative forms of the same gene, okay, these alternative forms of the same gene are known as alleles. But we should also note that in genetics, in most cases, they usually use these words gene and allele interchangeably, okay? So in a place where you're supposed to have an allele, they can easily use a gene. And where you have a gene, they'll easily use an allele. So they are usually used alternatively. But we should know the difference between the two, okay? Then we have a genotype. So the genotype is the genetic composition of an organism. Let's still take the example of Mendelian crosses with pea plants, okay? So you had two genotypes for someone who is tall, okay? Either you had this genotype, heterozygous genotype, or you had capital T, or you had this homozygous genotype. So in Mendel's crosses, you had two genotypes for tallness. Either you had this or you had this. Meanwhile, for shortness, you had just one genotype. That was T, small t. Okay? That is the homozygous genotype for someone or for a pea plant that was short. Meanwhile, for a pea plant that was tall, we had two genotypes. Okay, that is capital the heterozygote genotype for tallness and the homozygote genotype for tallness. Then, phenotype is actually the observable characteristic of an individual or the outward appearance. Okay, with Mendel's experiment, a pea plant was either tall Okay, a pea plant was either tall or it was short. There were no intermediaries. Okay, so in Mendel's experiment, we had two phenotypes. You were either tall or the pea plant was either tall or it was short. So these are things that you can see physically. So that is why they say the phenotype is observable characteristics. When a pea plant is tall, you see it as tall. When it is short, you see it as short. Okay. So we move now into disease inheritance. What is actually disease inheritance? These are diseases that are transferred from one parent or that are transferred from the parents to their offsprings through the gene. Okay, so these are known as diseases that are inherited. So there are two types of disease inheritance. We have the Mendelian disease inheritance and the non-Mendelian disease inheritance. But we are going to start with the different types of Mendelian disease inheritance that we have. Mendelian disease inheritance, these are disease inheritance that follow a regular pattern. That is, they actually follow the principles that were laid down by Mendel. Okay? In other words, from the genotype of the parents, you can easily say the proportion of the offsprings that will retain the genotype of the parents and those that will not. Okay, so it actually follows a regular pattern. Okay, and the first one that we'll be seeing is Mendelian disease inheritance is the autosomal dominant disease inheritance. These are inheritance carried on the non-sex chromosomes or autosomes, which require a single copy of the mutated allele for the individual to express the disease. An example here is the Huntington's disease. We are going to see this Huntington disease in front when we'll be looking at trinucleotide disease or repeat expansion. Okay, so when defining this Mendelian disease inheritance, there are things that we have to note that which are very important. If we have to define the autosomal dominant disease inheritance, we have to bring out the aspect or the position or the type of chromosome that carries the alleles. Okay, so we see here they are the non-sex chromosomes that carry the allele, and we also have to bring out the aspect of dominance. Okay, so that is why we say here that a single copy of the allele of the mutated allele will actually cause the individual to be sick. Okay, so for that definition to be complete, we ought to bring out those two aspects. Okay, in other words, since they are not carried on the non-sex, they are carried on the non-sex chromosomes or the autosomes. That means that the sex of the individual does not influence the distribution of this type of disease inheritance. Okay, whether you are male or a female, you people have an equal chance of having the disease. The male does not have it more than the female, or the female does not likely have it more than the male. Everybody has it an equal chance of having it. Let's take the example of a cross between an individual who has the Huntington disease. Okay, we have said it is autosomal dominant, so this person is sick with the Huntington's disease because he has one H, and the presence of one copy of this allele will cause this person to be sick. Okay, so this person, we have a cross between a sick person who is heterozygously sick and a normal person. Okay, so this individual is normal, so we have small h because a normal individual is this normal allele is recessive to the Huntington's allele, okay, which is dominant. So if we have a cross between a heterozygously sick individual and a normal individual, we are going to see that our phenotypic ratio of sick to normal will be 1:1. Okay, if we look here, we see that we'll have two offsprings that are sick, and we'll have two offsprings that are normal. So the phenotypic ratio of sick to normal is 1:1, while the genotypic ratio of those who are heterozygously sick to those who are normal is also 1:1. Okay, because we see these two are sick and these two are normal. Okay, now the second type of Mendelian disease inheritance is the autosomal recessive disease inheritance. This inheritance carried on the non-sex chromosomes or the autosomes, which require two copies of the mutated allele, okay, for the individual to express the disease. An example here, we have sickle cell anemia. Okay, so we require two copies of the mutated allele in order for the individual to express the disease because it is recessive. Okay, and they are carried on the non-sex chromosomes or the autosomes. Let's take this example: a cross between a carrier or a heterozygously normal individual and a heterozygously normal individual. Okay, so a cross between these two, the cross of the parent is generally known as the parent generation, while the first offsprings are known as the first filial generation. A cross between the first filial generation will give rise to the second filial generation. Okay, so this cross will give us 1/4 of the offsprings being homozygously normal. Half of the offsprings will be carriers or heterozygously normal, while one, okay, of the offsprings is sick. Okay, so you see that a cross between two normal people gives rise to the possibility of one individual being sick. You have two copies of the allele in this individual. Okay, so the phenotypic ratio of normal to sick is 3:1. Okay, this is 3 because we have one individual that we have this one individual who is homozygously normal, and two of the individuals are carriers. Okay, so they are heterozygously normal, while we have just one individual who is sickle cell. Okay, so we see that the phenotypic ratio of normal to sick is 3:1, while the genotypic ratio of those who are homozygously normal to carriers or heterozygously normal to the sick individual is 1:2:1. Okay, so this test of sickle cell anemia is one of the tests that is very important when you have to get married, okay, or you want to have a child with your partner. We have seen in this example that these two individuals are normal, but they have the chance of giving birth to a child that has sickle cell. Okay, so when you have to have a child or get married to your partner, you have to go to the hospital and do this sickle cell test. It is usually known as electrophoresis. So they will do electrophoresis and then they will tell you if both of you are carriers, you will be advised not to get married or to put an end to that relationship because there's a possibility that you people will give birth to a sickle cell child. Why should you get married that you have the possibility of giving birth to a child who will suffer with some sort of a disease? Okay, so the sickle cell test or electrophoresis is usually very important. One thing we also have to note here is that one excuse. The question that we'll be asking on our mind now is, why is it that we have sickle cell anemia that usually affects hemoglobin? Instead of putting H to start, capital H to start for hemoglobin, we are instead putting capital A to start for a normal hemoglobin and small s to start for sickle cell. The reason is that the protein that is generally affected in hemoglobin is alpha globin proteins. Okay, so this A stands for alpha globin. Okay, while S stands for sickle globin molecules. Okay, so normal hemoglobin with this normal alpha globin molecules will give normal hemoglobin in red blood cells with oval shape. Okay, so red blood cells like this one with normal hemoglobin are usually oval in shape. They are oval in shape and they are flexible. Okay, and this flexibility permits them to move across tiny blood vessels. Meanwhile, hemoglobin with a sickle shape gives red blood cell with sickle shape. Okay, so this is sickle hemoglobin, gives red blood cells with a sickle shape, and this sickle shape hemoglobin is usually stiff. They are not flexible, so it is difficult for this sickle hemoglobin to squeeze themselves across tiny blood vessels, so they usually accumulate like we see in this diagram. Okay, so they accumulate across blood vessels. When they accumulate, they prevent the transport of oxygen to neighboring cells around this area. Okay, to neighboring cells around this area. So they prevent the transmission of oxygen to these neighboring cells around this area, and they also prevent the transportation of carbon dioxide from neighboring cells back to the lungs. Okay, this generally leads to insufficient oxygen to be used in catabolism in cells to produce energy. That is why sickle cell patients generally have problems, energy crisis. Okay, generally have energy crisis. So what we are seeing here, we are actually trying to relate what takes place in the genome right up to what expresses itself phenotypically. Okay, now what causes this sickle cell is a mutation that transverse adenine. Okay, that transverse adenine to thymine. Okay, leading to the substitution of the amino acid valine to glutamic. So the substitution of the amino acid valine to glutamic acid. So this is what actually leads to the formation of this sickle hemoglobin. Okay, so it is a mutation that transfers adenine to thymine, leading to the substitution of this amino acid valine for glutamic acid. So the third example is autosomal incomplete dominance disease inheritance. This inheritance carried on the non-sex chromosomes in which both alleles produce an intermediary phenotype in the heterozygous genotype. Okay, and an example, we have the ABO blood group system in humans. One thing we should note here is that in the ABO blood group system, we have the existence of complete dominance and also the existence of incomplete dominance. Complete dominance is seen in the case where we have blood group A and blood group B, which are completely dominant over blood group O. Okay, and incomplete dominance or codominance is seen with blood group A and B that are incompletely dominant to each other, thus they produce an intermediary phenotype AB. So in incomplete dominance, none of the alleles actually is dominant over the other. Okay, so when they meet in the heterozygous genotype, they produce an intermediary phenotype which is different from that of A and is also slightly different from that of B. So phenotypically, what do we mean when we say someone has blood group A? Okay, so when they say someone has blood group A, is to say that this person has antigen A in his blood and anti B antibodies. Okay, so someone with blood group A, that is to say that the person has antigen A in his blood and anti B antibodies, while someone with blood group B has antigen B but anti A antibodies. Okay, anti A antibodies, while someone with blood group O means that the person has zero antigens. The person does not have either antigen A or B, so the person has zero antigens in his blood, but the person has both anti-B antibodies and anti-A antibodies. Okay, the person has both anti-A antibodies and anti-B antibodies. So the person has both antibodies for against antigen A and antigen B. Then someone with blood group AB has both antigen A and B, okay, but the person has zero antibodies. Okay, the person has zero antibodies. So how does this phenotype influence blood compatibility during blood transfusion? So what we are actually trying to do in this course is that we are looking what is taking place at the level of the genome and how that influences phenotypic expression, or how that influences gene expression, or how that influences the observable characteristics of individual. Okay, that is what we are trying to do in this course. So how does this genes or these alleles affect blood compatibility during blood transfusion? When we see this plus sign, is to say that it is compatible, okay? But when we see this red cross, is to say that it is not compatible. So from this table, we see that this line, this is for the donor's blood and this is the recipient's blood, okay? And from this table, we see that blood group O can donate to blood group O, blood group O can donate to blood group A, can donate to blood group B, can donate to blood group AB, okay? Blood group A can only donate to blood group A and can only donate to blood group A and to blood group AB, okay? Blood group B can only donate to blood group B and to blood group AB. Blood group AB can only donate to blood group AB. So blood group O is the universal donor because it can donate blood to everybody, while blood group AB is the universal recipient. You see, it can receive blood from everybody. Okay? Now, the very big question that comes to our minds is, how comes someone with blood group A will be able to donate blood to someone with blood group AB, but not vice versa? In other words, how can someone with blood group A be able to donate blood to someone with blood group AB, but someone with blood group AB cannot donate blood to someone with blood group A? Okay, the reason is that the factor that determines compatibility is actually the antibodies present in the recipient's blood, okay? the antibody present in the recipient's blood. We should note that very carefully. The antibodies that are present in the recipient's blood is what is the factor that determines whether the blood will coagulate or not. Okay, so let's take this our two examples. Someone with blood group A has antigen A, okay, and anti-B antibodies, okay, but someone with blood group AB does not have any antibodies in his blood, okay. So when someone with blood group A donates blood to someone with blood group AB, there are no antibodies in the blood of AB to cause this antigen A in blood group A to coagulate, in the donor's blood to coagulate, okay. But now, someone with blood group AB cannot donate blood to someone with blood group A, the reason being that the recipient who is blood group A has anti-B antibodies, okay. The recipient has anti-B antibodies, and these anti-B antibodies that are present in blood group A will cause the coagulation of this recipient's blood, okay, will cause the coagulation of the anti-B, will cause the coagulation of the antigens that are present in the will cause the coagulation of the B antigens, the B antigens that are present in blood group AB, okay. So when blood group AB donates to A, the anti-B antibodies that are present in the recipient's blood group A will cause the coagulation of will react with the antigen B that is present in the donor's blood, okay. So that is an explanation of what actually happens during blood transfusion. So the determining factor are the antibodies that are present in the recipient's blood. So the fourth example is gonosomal dominant disease inheritance. Mind you, we are still talking about Mendelian disease inheritance. This gonosomal dominant disease inheritance can also be called X-linked, okay, X-linked. So they are carried on the sex chromosomes. X-linked can also be called X-linked disease, a dominant disease inheritance. Gonosomal dominant disease inheritance can also be referred to as X-linked dominant disease inheritance. These are inheritance carried on the sex chromosomes. So here they are not carried on the autosomes. They are carried actually on the sex chromosomes, which require a single copy, okay, of the mutated allele for the individual to express the disease. An example is the Fragile X syndrome. So one thing we note with this gonosomal dominant disease inheritance is that the alleles are carried on the sex chromosome. That means that the sex of the individual actually influences the distribution of the mutated allele. Okay, so it influences the distribution of the mutated allele. So when you are a male or you are a female, you have a different probability of having the disease, okay. And one thing we also have to note with the gonosomal dominant disease inheritance is that fathers will only transmit this disease to their daughters, okay. Fathers will never transmit the disease to their sons. They only transmit it to their daughters, and they will never transmit it to their sons. Let's take this example, okay. This is a cross between a female or a woman who is sick, because she is sick with Fragile X. So this capital F stands for, it is a gonosomal dominant allele. So the presence of one of the allele will cause this woman to be sick, since it is a dominant allele. So we have a cross between a heterozygously sick female, okay, and a normal male. A normal male, okay, because he has small f. Since the normal f is recessive and the fragile X syndrome is dominant. So this man is normal, okay? So when we do this cross, we will see that the phenotypic ratio of normal to sick is 1:1, okay? You see this woman and this man, they are sick, and this woman and this man, they are normal, okay? So the phenotypic ratio of normal to sick is 1:1. While the genotypic ratio of this heterozygously sick woman to a homozygously normal, or to a normal woman to a sick male to a normal male is 1:1:1:1. Okay, so we have 4 genotypes. Okay, they are all different. They are all different here because they are carried on the sex chromosome. Okay, so the fifth is the gonosomal recessive disease inheritance. These are inheritance carried on the sex chromosomes, which require a single copy of the mutated allele in a male, or both copies of the mutated allele in a female for the disease to be expressed. And an example here, we have hemophilia. Hemophilia is actually a disease state that there is difficulties for blood to clot. Okay, there's difficulties for blood clotting. Now we see here that we said that with gonosomal, they are actually carried on, the alleles are actually carried on the sex chromosomes, okay? And that means that sex actually affects the way, the proportion, or the distribution of the alleles. We have seen here that a single copy of the mutated allele is required for a male to be affected, to have the disease. But in a female, we need both copies to be mutated, since it is recessive, okay? And the reason is because males have only one X chromosomes, okay? They have only one X chromosome. So if they have just one copy of this hemophilia that is carried on this X chromosome, there is no other X that will carry a normal allele that will produce the normal proteins, okay? So automatically, if this X is carrying one mutated allele, this male will be sick, since the Y chromosomes do not carry alleles, okay? But in a female who carries two X chromosomes, okay, if one of them is sick, if one of them is sick, okay, if one of them is sick, let me put some more S. If one of them is sick and or is mutated, and the other one is carrying a normal allele, this normal allele will produce the normal proteins, okay? Which, since it is recessive, that normal proteins will overshadow the mutated allele, and so the woman will not have the disease. But if the woman has to have the disease, since it is recessive, she is supposed to carry two sick, okay? She's supposed to carry two sick or two mutated alleles, okay, in order for the disease to be expressed. So we see here that the sex of the individual, or the gender of the individual, is actually influencing the distribution of the disease. And because this is happening, male will more likely have the disease than females, okay? Because male, they only have just one X chromosome. So if one is mutated, they automatically have the disease, okay? And one peculiarity with gonosomal recessive disease inheritance also is that fathers will only transmit it to their daughters, okay? They cannot transmit this type of disease inheritance to their sons, okay? Since it is transmitted on the X chromosome, they will only be transmitting it to their daughters and not to their. So the sixth Mendelian disease inheritance is the Y-linked disease inheritance. These are disease inheritance carried on the Y chromosomes. Okay, usually Y chromosomes do not carry genes except for the sex determining gene carried on the sex determining region Y, usually abbreviated as SRY. Okay, an example is the Y chromosome in fertility. Okay, and one thing we should know with the Y-linked disease inheritance, one peculiarity with it is that fathers do not transmit it to their daughters. They only transmit such an inheritance to their sons. Okay, fathers only transmit it to their sons since daughters do not have Y chromosomes. So a father can only transmit it to the son. Now, the very big question is, how can a father who has the Y chromosome infertility, that means that he is infertile, how can he transmit this infertility to the son? If he is infertile, that means that he will not be. How will he be able to produce a child? Not to talk of transmitting the Y chromosome infertility to the son. Okay, so that is the very big question that should be on our mind. If fathers only transmit it to their son, how will a father who has the Y chromosome infertility be able to bear a child, not to talk of transmitting it to the son? Now, in order to answer this question, we have to define infertility. Okay, infertility can be defined as the inability of an individual to reproduce a child through the normal biological process, that is through sexual intercourse, the normal biological process. Okay, now that we have defined it, we can now answer the question. Okay, so we have three scenarios that are possible. In one of them, the father may just have a predisposition for the Y chromosome infertility. Now, if the father has the predisposition, it might be due to the fact that we have epigenetic factors that are actually preventing this predisposition to express itself in the father. Okay, now when it is transmitted to the offspring, it will actually multiply itself and then express this Y chromosome infertility. Okay, that would simply mean that the son actually inherited it from the father, although the father did not. It was not expressed phenotypically, but he transmitted it to the son, and now it expresses itself phenotypically in the son. That is the first scenario. We said what can cause this might be due to epigenetic factors. Okay, we are going to see epigenetic factors under non-Mendelian disease inheritance. The second scenario is that the father may be producing non-motile sperms. Okay, if the father is producing non-motile sperms, the father can be helped, okay, through assisted reproduction. Okay, if the father is producing non-motile sperms, that means that he produces sperms. These sperms will not be able to swim to the oviduct, okay, to meet the egg cell of the wife and fertilize it to produce offspring. Okay, so the father will be infertile. Okay, and if this happens, the father can be given assisted reproduction because the sperms can be extracted, okay, and then used to fertilize the egg cell of the wife through artificial insemination or assisted reproduction. And if that happens, that father's Y chromosome infertility will also be transmitted to the son. Okay, will also, if this couple happen to bear a child, or to give birth to a child that is a son, that means that the father's Y chromosome infertility that led the father to be producing non-motile sperms will also be transmitted to the son. Okay. In a third scenario, the father may have low sperm count, and if he has low sperm count due to the Y chromosome infertility, he might not be able to fertilize the egg or the egg cells of the wife through the normal biological process. And in this case, he can also be helped through assisted reproduction. Okay, the sperm cells can be taken and then artificial insemination used to fertilize the egg cell of the wife. And if that happens and they give birth to a son, that Y chromosome infertility that was present in the father will also be transmitted to the son. Okay? So the seventh one is Mendelian cytogenetics. What is cytogenetics in the first place? It is the study of chromosomes. It is the study of chromosomes and related abnormalities. Some abnormalities or mutations develop in chromosomes and are transmitted to the offsprings in a Mendelian manner. Okay, so that is why we have also brought this cytogenetics under a form of Mendelian disease inheritance, because the chromosome might have a mutation and then it is transmitted in a Mendelian manner following a regular pattern to the offspring. Okay, mutations might either develop spontaneously. That means that it is not induced. Okay, and an example is the case of where we have dysfunctioning of or mistakes that are made during DNA replication, during DNA repairs. Okay, when replication takes place, an error might occur during DNA repairs. If that happens, then it will develop into a mutation, and that is a spontaneous means to which mutation may develop. Mutation might also develop artificially. That means it is actually induced. It might be induced by humans in the lab, or it might be induced by other substances or by radiation such as X-ray radiation. Okay, now there are three types of mutation. Okay, the first one is genomic mutation, which is actually due to a change in the chromosome number. This genomic mutation is divided into two. We have the one that results in the change in the number of chromosomes, which usually leads to the formation of aneuploidy. An example of an aneuploidy is trisomy, that is 2n plus 1, or monosomy, 2n minus 1. And we also have a change in the chromosome set, which leads to the formation of euploidy. Okay, and an example of a euploid cell is a triploidy, that is 3n, and a tetraploidy, that is 4n. We see that for euploidy there is a change in the chromosome set, while for aneuploidy there is a change in the number of chromosomes. Okay, the second type of mutation is chromosomal mutation. This is actually a change in the structure of the chromosome. Okay, there's a change in the structure of the chromosome, and it is then transmitted to the offspring through a Mendelian manner. And an example of such chromosomal mutation, we have chromosomal deletion. That means a portion on the chromosome is actually deleted. Okay, and this deleted chromosome can now be transmitted to this chromosome that has lost a portion of its length can then be transmitted to the offspring. It can be a duplication. That means a section on the chromosome actually doubles itself. Okay, it can be an insertion, it can be a translocation, it can be an inversion. Okay, and the third one is gene mutation, which is a change in the gene nucleotides. Okay, an example here is the gene nucleotide deletions, gene nucleotide translocation, duplication, and inversions. So here we are going to see an example. We are still under Mendelian cytogenetics, an example for the formation of a euploid cell. I will also see an example for the formation of an aneuploid cell. We know during meiosis, the purpose of meiosis is usually actually to reduce the 2n number of chromosomes or the diploid number into half the number of chromosomes, that is into n plus n. So this is the haploid number, the 2n, and here we have the haploid number, that is n, or half, with half the number of chromosomes. So that is the purpose of meiosis. So cells undergo meiosis so that they can reduce this diploid number into a haploid number, usually in gametes. Okay, now if there's a meiotic error like what we have here on the board, so we see here that the 2n here is equals to 6. 1, 2, 3, 4, 5, 6. So the 2n number here is, the diploid number here is equals to 6. So this is supposed to reduce to n, that is equal to 3, to 2 cells of n equals to 3, to and n plus n equals to 3 at the end of meiosis 2. Okay, but now there's a meiotic error here such that that 2n is not reduced, and now we produce a gamete with still that retains the diploid number, that is 2n here, still equals to 6, and 2n here still equals to 6. Okay, so during self-fertilization, this will combine again to give a cell with 2n plus 2n equals to 4n, that is equals to 12. Okay, so we'll have an abnormal cell that is formed here, and this cell is an euploidy. Yes, a euploidy, a tetraploid. Okay, because there's a change in the chromosome set. Okay, also this 2n here can fuse with a normal n cell. Okay, it can fuse with a normal haploid cell to give a cell with a 3n, which is a triploid. Okay, so this 2n, this abnormal 2n, can still fuse with a normal haploid cell to give a 3n, which is a triploid. So that is the formation of a euploid cell. And here it is, we have two examples: tetraploid, the formation of a tetraploid, and the formation of a triploid. Okay, so let's come now to the formation of a euploid cell. Okay, during meiosis, okay, that is meiosis 2. Now here we have meiosis 1 that has given rise to this cell. We have meiosis 2, which is supposed to give us 2 cells of n plus n. Okay, but here there is a non-disjunction. Okay, there is a non-disjunction at this level. There's a non-disjunction here. Normally during meiosis 2, there is the separation of the daughter chromatid. Okay, like here, this one takes place normal. Okay, you see this other daughter chromatid is going to this side, while the other one is going to the other cell. But here, okay, the two are moving in one direction. The two are moving in one direction. Okay, that is a non-disjunction. A non-disjunction has taken place during meiosis 2. This will lead to a cell that has n plus 1. Okay, n plus 1. And this other one that will lose a daughter chromatid will have n minus 1. Okay, now when non-disjunction has taken place, giving rise to an abnormal cell n plus 1 and n minus 1, okay, this n plus 1 can fuse with a normal haploid cell to give rise to a cell, an abnormal cell, that is 2n plus 1, okay, which is known as a trisomy. This is a trisomy. Okay, or we may have a case where it is this other one, this other abnormal cell n minus 1, fusing with a normal haploid cell, okay, a normal haploid cell to give a monosomy, that is 2n minus 1, okay, and these are different forms of aneuploid or aneuploidy, okay. So we are done with Mendelian disease inheritance. We move now into the second disease inheritance, that is the non-Mendelian disease inheritance. These are disease inheritance that do not follow a regular pattern. That is, they do not follow the principles that were laid down by Mendel. In other words, from the genotype of the offsprings or from the genotypes of the parents, you cannot say the proportion of the offsprings that will retain the genotype of the parents and those that will not. So it does not follow a regular pattern, okay? And the first example we'll be looking here is the trinucleotide repeat disorder. These are disorders brought about by the expansion of triplets of bases above a certain threshold. An example is the Huntington's disease. Remember when we were looking at autosomal dominant disease inheritance, I said we are going to look at the Huntington's disease under trinucleotide repeat disorder. So this is it we are seeing here, okay? We have an example, the Huntington's disease, that is brought about by the expansion of this triplet CAG. Okay, this is a triplet of bases. Okay, so it is caused by DNA slippage. Okay, so let's try to explain this for using the figure below. Let's consider that this is a double strand of DNA. Okay, this is a double strand of DNA with the upper strand and the lower strand. We know during DNA replication, okay, the enzyme helicase, which we have represented here by this small spherical green, this enzyme helicase has to unfold this DNA double strand, okay? So, in the course of unfolding this DNA double strand here, okay, this helicase forms a loop, okay? It pulls this, you should note these triplets that I've put in blue, okay? These are the ones that have been pulled up here. Okay? So helicase pulls them up and then forms a loop. Why is it able to form a loop? It's because these triplets are actually repeating themselves, okay? Since they are repeating themselves, when these two triplets are pulled upward, okay, you see that this other triplet will come and replace this first one, will slip, okay, and then bind and then pair up with this other triplet down here, okay? So this triplet here, let me name it number 2, or let me name it number, yes, this triplet number, let me name it number 4. This is it down here. This is the triplet number 4, okay? That has now paired up with the second triplet of the second DNA strand below, okay? Now, when this happened, when DNA polymerase, which we have figuratively presented it here as this yellow spherical ball, when DNA polymerase now comes in to polymerize the chains, okay, or to form the other strand, okay, it will come and then put bases here like that and go up and come down, put bases here, okay, put bases on this strand below. When it put bases on this strand, it is going to form a strand with that has two triplets more than the original double strand DNA, while this lower strand will give a shorter double stranded DNA. Okay, so we see that there is a disequilibrium, because this one has two triplet plus, while this one does not have any additional two triplet. And this is because of DNA slippage that has been caused by helicase that formed a loop here, okay? And because the triplets are repeating, even this base here can come and pair with this base and there will be no problem, okay? So we notice here that we have a strand. This strand here is two triplets longer than the original DNA double strand, while this other double strand has the same length like the original DNA double strand, okay? So we'll see that this will keep on repeating itself, this will keep on repeating itself, and this one will keep on producing, adding triplets and adding triplets until it reaches a certain threshold. And then when it reaches a certain threshold, the trinucleotide repeat disorder will happen, okay? And here we generally talk about genetic anticipation. That is, the disease keeps on worsening up as we move from one generation to the other, okay? The disease keeps worsening up as we move from one generation to the other. One thing we should note here is that these triplets actually occur normally on the DNA, okay? They actually occur normally on the DNA. So these are the triplets that are even usually exploited for DNA fingerprinting. These repeats are normally on the DNA. But now, trinucleotide repeat disorder happens when this triplet goes above a certain threshold. Because when it goes above a certain threshold, it becomes a disorder, okay? So the second non-Mendelian inheritance we'll be looking at is epigenetic inheritance. Epigenetic inheritance involves factors which influence gene expression without affecting the DNA sequence. Okay, an example here we have methylation, acetylation, and microRNAs. Okay, let's look at this figure below. This figure actually shows a chromosome. Okay, this is a chromosome. Okay, when you unwind a chromosome, you will see that you have this portion, okay, that is a heterochromatin, where the chromosome, the DNA is actually compacted, okay, so it cannot express itself because it is compacted, okay. And also when you unwind a chromosome, you have a portion that is open or loose. This portion is known as the euchromatin, okay? And in this position, the DNA can actually express itself. So transcription can take place when the DNA is in the form of the heterochromatin. But when it is in the form of when it is the form of euchromatin, when the DNA is in the form of euchromatin, DNA transcription can take place. But when the chromosome is in the form of the heterochromatin, the compacted form, DNA transcription cannot take place. In other words, gene expression cannot take place. Okay. Now on this round stuff here are proteins known as histones. Okay, known as histones. These are proteins known as histones. Okay, while this rope-like stuff is the that winds around those round stuff is the DNA. Okay, is the DNA. So how does this epigenetic factors influence gene expression without affecting the DNA sequence? What happens is that methyl, or the presence of methyl group, is methylation. Okay, so the methyl groups are actually hydrophobic, so they will favor the DNA, they will cause the DNA to shift towards this configuration, okay, or to shift towards this form of heterochromatin. And now when DNA is shifted towards this form of heterochromatin, gene expression cannot take place. Okay, so that is an influence on gene expression by methylation. Okay, on the contrary, acetylation is hydrophilic, so it will push the DNA to express itself towards this configuration of euchromatin, okay, will cause the DNA to express itself in this form of euchromatin. And when the DNA is now in this configuration, gene expression can take place, transcription can take place, okay. Now, microRNA, how do they influence gene expression? MicroRNAs usually bind to messenger RNAs and therefore inhibit the messenger RNA from being translated. Okay, so you see that all of these epigenetic inheritance or factors, they will influence gene expression without actually affecting the DNA sequence. So the third one, non-Mendelian inheritance, is the genomic imprinting. Okay, this is a heritable disorder caused by natural silencing of an allele by epigenetic factors. An example here we have the Angelman syndrome, in which the maternal imprinted or silenced, the maternal allele is imprinted or silenced in the offspring, and only the paternal allele, which is a mutant, is expressed. Okay, genomic imprinting is actually the silencing of an allele such that the allele cannot express itself. Okay, so that is a form of a non-Mendelian disease inheritance. Okay, and here we have given the example of Angelman syndrome. What happens during Angelman syndrome is that we know that each individual for a particular gene has two alleles. Okay, one of those alleles comes from the father and the other allele comes from the mother. Okay, in Angelman syndrome, the maternal allele is actually imprinted. In other words, it is silenced. Now, since the maternal allele is silenced, there is no problem at this level. The father's allele can still express itself normally. But what happens in Angelman syndrome is that the maternal allele is imprinted. Then instead of the paternal allele to express itself, the paternal allele is mutated. And so it expresses a mutant allele, okay? So it will express an abnormal protein, okay? Which will now lead to the Angelman syndrome, okay? So the question now that we have to answer is, what is therefore the difference between a recessive allele and an imprinted allele? A recessive allele and an imprinted allele. The difference between a recessive allele and an imprinted allele is that a recessive allele will express itself in the homozygous form, but an imprinted allele, even in the homozygous form, will not express itself because it has been imprinted, it has been silenced. Okay, so even you double two silenced alleles and put them together, it will not express itself. So that is a difference between a recessive allele and an imprinted allele. So the fourth non-Mendelian disease inheritance is the extranuclear inheritance. This is inheritance of extranuclear or non-chromosomal DNAs, such as mitochondria and chloroplasmic DNAs. Okay, an example we have in humans, children only inherit mitochondrial DNA of their mothers found in the egg cell and known from their fathers. Okay, so extranuclear inheritance is actually the inheritance that is carried in the non-chromosomal or the non-nuclear DNA. Okay, and here we have cited the mitochondria in humans and the chloroplast DNA, the mitochondrial DNA in humans and the chloroplast DNA in plants. Okay, so the mitochondria has its own DNA. Okay, the chloroplast also has its own DNA. So what we are talking about extranuclear inheritance are the inheritance of DNA that is not carried on the chromosome, or DNA that is out of the chromosome. Okay, so it's the transmission of mitochondrial DNA and chloroplasmic DNA, non-chromosomal DNAs. Okay, and here we have talked about the case of humans. In humans, during fertilization, we know that the egg cell usually have its own mitochondria. Okay, The female egg cell usually have its own mitochondria. But now the sperm cells, okay, they also have their own mitochondria, but that mitochondria is found in the flagella, okay? So this is the sperm cell with its own mitochondria, and the egg cell with its own mitochondria, okay? So during fertilization, only the nucleus of the sperm cell usually moves into the egg cell, okay, while the flagella of the sperm cell is disposed of, okay. So it is disposed of. That means that the mitochondria that is found in the flagella is also disposed of, okay. When it is disposed of, it will not fuse, it will not move into the egg cell. That means that the mitochondria of any child, the mitochondria of any child comes from the mother, because now when the nucleus of the sperm cell fuses with the egg cell, it produces a zygote that later develops to the embryo, okay. And you will see that the mitochondria that will be present in the zygote or the embryo will be only the mitochondria that was present in this egg cell of the mother, this egg cell of the mother, okay. So that is an extranuclear inheritance. If they want to determine your maternal line, a pure maternal line, they will have to come and extract the mitochondrial DNA of each individual. Every individual's mitochondrial DNA comes from his mother, okay? It's only from the mother and not from the father. Now, the extranuclear inheritance can be of three types. We have the uniparental, which is just the case that we have mentioned here in humans, okay? Just one parent supplies the extranuclear DNA or the non-chromosomal DNA. We have biparental, okay? This, we can see it. In this case, both parents supply the non-chromosomal DNA. And an example of this case is sexual reproduction by conjugation in yeast cell, okay? Sexual reproduction by conjugation in yeast cell. Here we see where both hyphas, both hyphae, they grow towards each other, and at a certain point where they meet, they fuse their cytoplasm. Where they fuse their cytoplasm like that, you have extranuclear DNA inside, and that place that the cytoplasm fuses develops into a zygote. That later develops into a zygote spore, okay? So we see that the hyphae, which one is denoted as positive, the other negative, will supply the non-chromosomal DNA to the offspring, okay. And the last one is vegetative segregation, okay. And then this one, we have a cytoplasm, okay. We have the non-chromosomal DNA that is distributed in a non-equal, okay, is distributed in a non-equal form in the offspring, okay. And an example here is sexual reproduction, no, asexual reproduction by budding in yeast, okay. Asexual reproduction by budding in yeast. So you see that each of these buds has an unequal proportion of the non-chromosomal DNA, because the cytoplasm divides in an or in a non-regular form, okay, to provide. So you might have this with 4 mitochondria, this other cell with 5 mitochondria, this one with and so on and so forth. So you have a disequilibrium in the distribution of the non-chromosomal DNA. Now we have dosage compensation, which is also known as leonization. Leonization comes from the name of the person who discovered dosage compensation. Her name was Leon. Okay, so dosage compensation, also known as leonization, is the process of inactivating one of the X chromosome in somatic cells of females in order to equalize the dose, in order to equalize it to the normal dose, in order to equalize it to the normal dose. Let me erase this. In order to equalize it to the normal dose of one X chromosome found in males. Okay, so generally human males have one X chromosome in their somatic cells while females have two X chromosomes. The question now is, who is the one who has the normal dosage of chromosomes? Okay, so the person with the normal dosage of chromosomes are males, okay, because they have one X chromosome, and this one X chromosome can provide all the, or can carry all the alleles that are needed, okay, and can express itself to produce all the normal proteins that are needed. So females with two X chromosomes actually have an overdose of X chromosomes. So since they have an overdose, something has to be done to reduce this overdose so that it does not cause abnormalities, okay? And that is generally done through the process of dosage compensation, or X chromosome inactivation, or the process known as leonization, okay? That usually happens by one of the X chromosome in the female somatic cell being condensed into a heterochromatid. Okay, one of the X chromosome in the female somatic cell is condensed into a heterochromatid, which is called a bar body. Okay, this bar body is there, but it is not functional. So that will allow just one X chromosome in the female somatic cell to express itself. We should note that this does not take place in the gamete, okay? Because in the gamete we have just one X chromosome. Okay, so it takes place in the somatic cells where we have, for somatic cells of female, where we have two X chromosomes, because in the gamete of females we have, they have already been reduced to half the number of chromosomes, so we will have an X cell in the gamete and an X cell. So these gametes with one X and one X do not need to a dosage compensation because they already have the normal dose. But when it is the somatic cell that of females where we have two X chromosomes, okay, dosage compensation has to take place to condense one of them into a heterochromatid that is called a bar body. Let's look at the examples in this table. We have a female somatic cell with two X, okay. What happens is that one of them is condensed into this bar body while the only one is active, okay. It is there, but it is existing as a bar body, okay. We have in male somatic cells, we see that males have the normal dosage 1, so there is no need for the formation of a bar body. Now we have the Klinefelter syndrome, okay. The Klinefelter syndrome, we see that these are males with two X chromosomes. So since the males, they have two X chromosomes, one of it has to be condensed into a bar body, okay. And we have the triple X syndrome. Those are females with 3X. So these females with 3X, they have to condense two of the other X, to form these two bar bodies, okay, to form two bar bodies so that only one of the X chromosomes is active. Okay, we are done with disease inheritance, and now we move into evolution. Evolution, what is it? This is one of the most controversial topics in biological science, okay. Some people have gone as far as believing that those who even teach evolution, they do not actually believe in creation or the existence of God. But I would differ with them on this idea, because evolution is even one of the ideas that actually tries to support the existence of God. We'll see that when we look at the different theories, okay, about evolution. Evolution and creation, it is just is we can link it to the analogy of an individual coming into a class with a yellow orange fruit, okay? If I come into this class with a yellow orange fruit and I ask you a question, is this yellow or is this a fruit? What will be your answer? If I come with a yellow orange fruit and ask you a question, is this yellow or is this a fruit? What will be your answer? So you see that my question is actually prompting you to make an error because both of the answers are correct, but my question wants that you should choose just one correct one. How can you choose between two correct answers? Okay, it is difficult. So that is the same thing with evolution and creation. Okay, it is not because creation took place, which means that evolution cannot take place. Or it is not because evolution is taking place, which means that creation did not take place. Okay? Evolution and creation, me, I see it as complementing each other and not actually contradicting each other. Okay? So we should have that in mind. Okay? Evolution is not saying that creation did not take place. Neither is creation saying that evolution cannot take place. Okay? So you should keep that analogy of a yellow orange fruit in your mind. It is very important. So what is there for evolution? Evolution is actually a change of genetic frequency in a gene pool, okay? Or the change in genetic frequency in a population over a given period of time. I've talked about gene pool. Gene pool is actually the total genes that are found in a population within a given time, okay? So either we define evolution as a change in the genetic frequency in a population over a given period of time, or a change in the genetic frequency in the gene pool over a given period of time. We cannot talk about evolution without talking about life and the origin of life. What is life? I bet you life, even scientists will confess that they have found it very difficult to define the word life. So they have defined life by actually combining the characteristics of living things. So life is defined as the ability of an individual or of anything to be able to respire, to be able to reproduce, to be able to respond to a stimulus, or and to be able to fit. So that is the definition of life. So you see, it is a collection of the characteristics of living things. So if you think that life is difficult to define, then I bet you know that the origin of life is even far more difficult. Okay? So there are many theories that have been put forth to try to explain the origin of life. And some of these we have the theory of spontaneous generation. According to this theory, life originated from non-living thing. Okay? According to the theory of spontaneous generation, life originated from non-living thing. But Pasteur actually disproved this by using his experiments with spores. Okay? So he disproved this by saying that living thing can only originate from a living thing and not from a non-living thing. Okay. The second one, we have the theory of spatial creation or scientific creationism. Okay. This is actually the theory on which many religions are based. According to this theory, life on earth has been created by a superior being which they call God. Okay, that is according to the theory of spatial creation. That means there is a superior being which they call God who actually created life here on earth. But some scientists have argued that they cannot accept this theory. Reason being that you cannot just sit and accept that somebody somewhere created life here on earth without proving it scientifically, or using experiment to prove that it actually happened. So according to them, they disagree with the theory of spatial creation based on that. Okay, we have the third one, which is the Oparin-Haldane hypothesis. According to this theory, or according to this hypothesis, lightning may have provided the spark needed for the generation of amino acids, sugars from water, methane, ammonia, and hydrogen molecule. Okay, now the question is, why is it that lightning today is no longer leading to the production of life? Okay, although they try to argue that at that time the environment was too harsh, there was a high temperature, there was high lightning and high volcanoes that could have make the difference for lightning to form life at that time, as compared to now that the environmental conditions are not too harsh. Okay, now we have the RNA wall hypothesis. According to the RNA wall hypothesis, okay, we know that we need DNA in order to synthesize proteins, okay, and we need proteins in order to synthesize DNA. So, which one first appeared here on Earth to form life? Okay, because if DNA was the primary molecule, it means that it will obviously need proteins in the form of enzyme in order for life to be formed. Okay, then if it was proteins will obviously need DNA in order to be synthesized. So according to the theory of RNA wall hypothesis, it says that RNA should have likely been the molecule that originally existed, and then from this molecule, since RNA can both function as an enzyme and as a nucleic acid material, okay, a nucleic acid material from which life should have originated. Okay, but now the question still lies, how did RNA come here on Earth? Where did that RNA come from? So you see that that also has a limit. Okay, now we have the fifth hypothesis, is the protein wall hypothesis. Okay, the protein wall hypothesis. According to this hypothesis, life may have originated in a warm water pond, okay, loaded with all sorts of ammonia, phosphorus salts, light, heat, and electricity. The question is, why is it that today we still do not have such life being formed? Okay, and this proton wall hypothesis was set forth by Dalton. Okay, and he tried to argue that in the past there was no life, so these molecules could actually form like that. But today, in our days, we have plants, so if these molecules should form life in a pond like that, they want to form life in a pond like that, the plants easily pick them up and use them as nutrients. Okay, but that is not still tangible. It does not still give us where life originated here on Earth. Okay, now we have the theory of panspermia. Okay, the theory of panspermia. According to this theory, life may have originated from outer space and then brought to Earth by cosmic blast. So according to the theory of panspermia, life should have originated somewhere else, and then due to cosmic blast, this should have been carried to Earth. But if this be the case, then the question would just simply change to where life originated out of Earth. Okay, so this does not still justify the origin of life in that outer space before it was brought here on Earth. Okay, so we move now into the evidence of evolution. What are some of the proof that might try to show that evolution is taking place? The first proof, we have the the universal genetic code, the existence of the universal genetic code, okay? The fact that all cells on earth can read any piece of DNA from any form of life on earth may be a proof that all life have a common ancestor, okay? Now, all cells on earth can read a piece of DNA no matter its origin. So this might try to tell us that means that all these organisms actually had a common ancestor. Okay, that is why their cells can read DNA from all of the diverse form of life that we have on earth. Okay, the second evidence is the fossil record. The fossil record shows that the fossils of the simplest organisms are found in older rocks, okay, while more complex fossils are found in more recent rocks, okay. That is trying to say that might be initially or originally we had just one simple ancestor, okay, from which different lives started evolving and producing more complex organisms. So the fossil records actually shows that at the bottom in the older rocks we have very simple organisms, but while we are moving up with the fossil record we have more complex organisms in new rocks, okay, more complex organisms in new rock. That means that organisms started might be from a single ancestor and then went up to produce diverse forms of lives, okay. Now we have genetic commonalities. Genetic commonalities. The similarities in genes from humans, apes, cats, cows, etc. may show that we had a common ancestor in the past. Okay? May show that we had a common ancestor. So the resemblance in these organisms, these different organisms, their DNA, okay, because their DNAs are they are similar. This might show that these organisms, cats, cows, apes, humans may have had a common ancestor that went on and later deviated to give the different lives that we have. This might be an evidence that evolution actually took place. Mind you, you should note this very well. These genetic commonalities does not say that humans and apes and cats are the same. Okay, or it does not say that humans should have originated from apes or from cats. It says that these organisms, humans, apes, may have had a common ancestor. Okay, so we should mind the difference between the two, because most people misinterpret evolution and they say that okay, it's like that we originated from apes or we originated from chimpanzees. No, it is saying that this similarity between humans and apes and cats and all the others might show that we had a common ancestor. So we should note that difference, okay? So we have common traits in embryos. This actually shows that there is the recapitulation of ancestral history, okay. Common traits in embryos. This actually shows the recapitulation of ancestral history. That is, an embryo goes through the developmental stages of its ancestors before being fully formed, okay. So each embryo goes through the ancestral history of its ancestors before actually forming its full embryo. This might actually show that these organisms originated from a common ancestor in the past, okay. And so we also have bacterial resistance. When we culture bacteria, we always say that the bacteria has developed resistance and so on. So when antibiotic is placed in a bacterial culture, only the resistant bacteria survive and multiply. This may be a proof that evolution that only the fit survive. Okay, that this might also be a proof that evolution takes place. Because we put an antibody into a culture, it kills those that are not resistant, while those that are resistant will multiply, okay, will multiply and then propagate their fit genes in the population. So that might also be a proof that evolution has taken. Now, the driving forces for evolution, we have natural selection. Okay, so natural selection, this was brought forth by Charles Darwin, okay, who said that there is survival of the fittest. So organisms that are fit will survive and propagate their fit genes in the population, while those that are not fit will die and be eliminated. Their genes will be eliminated from the population. We should note here that the notion of fitness does not mean the ability to produce a lot of offsprings, but fitness here ties to the ability of an individual, of an organism, to produce viable offsprings that can be able to produce other offsprings. Okay, let's take for example that we have two individuals here, okay, at the beginning, who mate and produce around 100 offsprings, and these 100 offsprings are not able to mate, are not viable to produce other offsprings, and they therefore produce zero offspring. And an individual, we have two here, okay, and the second case that we have two individuals who mate to produce around 4 offsprings, and these offsprings, they mate to produce around 8. Okay, so we see that this second case is more fit than the first case. Okay, that second case is more fit than this first case. We see that this first case has produced a lot of offsprings, 100 of them. Okay, but these offsprings are not viable, they cannot produce other offsprings, so this organism is not fit. Okay, why this one is fit, this one is well adapted, that is why it produces offsprings that can produce other offsprings. Okay, so that is the notion of fitness that Darwin, or Charles Darwin, was talking about. We have gene flow. This is the move of new genes into a population. So you have the introduction of new individuals into a population, that is known as gene flow, and that is a driving force for evolution as well. We have mutation. Okay, when you have a population of individuals and then there is mutation, it actually lead to a change in the genetic frequency. Okay, and that is also a driving force for evolution. We have genetic drift. This is actually a chance event or a catastrophe that tends to eliminate certain individuals from the population irrespective of their genotype. So it should be a chance or a random event, like a catastrophe, okay? You should not select individuals based on the genotype that they have, because if you start selecting individuals based on the genotype that they have, then it becomes natural selection. It is no longer genetic drift. So genetic drift is different from natural selection, in the sense that they, a random or a chanced event should eliminate certain individuals from the population, not depending on their genotype. It should eliminate them randomly. Okay, we have a case like a tsunami or an earthquake that comes and eliminates some people, and some people are left by chance, not because they have a particular genotype. Now, if we have something like sickle cell comes in and start eliminating those that have the sickle genotype. Okay, those that have the sickle genotype, that is no longer a genetic drift, it is natural selection. Okay, we also have selective reproduction. So we should actually, reproduction should be random. So we should not have some individuals with some particular traits that permits some mate to be attracted to them, such that those people, they propagate more of their genotype or of their genes in the gene pool. Okay, so reproduction should be non-selective, because when it is selective, then those that have a particular a trait that attracts more mates will have more of its genes propagated in the population, and that will lead to a change in the genetic frequency in that population. Now we have the Handy-Weisberg equilibrium. Handy-Weisberg equilibrium. Now when Mendel was doing his crosses with, or his experiments with the pea plant, he noticed that the genetic frequency was actually changing in his small pea plant population, and he did not have an explanation for that. Okay, so he was doing a cross with little population of pea plants, and so this was leading to a change in genetic frequency in that his small pea population. Okay, so Handy and Weisberg actually came with an equation with certain conditions to be fulfilled, okay, in order for a cross to respect this equilibrium. Okay, so they are the ones that actually notice what was the difference, or that actually explain what was actually causing the changes in the genetic frequency in Mendel's experiment. Okay, so what is the Handy-Weisberg equilibrium? The Handy-Weisberg is a situation where there is no change in the genetic frequency in a population, and like we said, it is an equilibrium, so there is no change in the genetic frequency in the population. We should notice that when Mendel was doing his crosses, you were either tall, or the pea plant was either tall or it was short. Okay, and the proportion of the offspring was supposed to sum up to that of the parent. In other words, he was using the phenotypic frequency. Okay, so P stands for the frequency, the frequency of the dominant allele. P stands for the frequency of the dominant phenotype. Okay, P stands for the frequency of the dominant phenotype, while Q stands for the frequency of the recessive phenotype. So this, we should say P stands for the tall plant, while Q stands for the short plant. Okay, so these phenotypes must sum up to 1. That is the first equation that Handy-Weisberg brought out. Okay, and now knowing that in each phenotype, okay, there is the existence of 2 alleles. Okay, so in each gene we have the existence of 2 alleles. They square the both side of this equation. Okay, they square the both side of the equation. So they square this, and they square this other side. Okay, that leads to this other equation. When you open this, you expatiate, or you expand this equation, it gives rise to equation 2. Okay, and in this equation 2, P square stands for the frequency of the homozygous dominant genotype. 2PQ stands for the frequency of the heterozygous genotype, while P square stands for the frequency of the homozygous recessive. We see that from equation 1, we could not determine, okay, the frequency of the heterozygous genotype. Okay, that is this 2PQ. And also we could not derive the frequency of the homozygous dominant genotype, and also we could not derive the frequency of the homozygous recessive genotype. But now when we square the both sides of the equation, from this equation 2, we can easily determine those frequencies. Okay, so these are the main equation 1, and equation 2. Equation 1 will permit us to determine the frequency of the dominant phenotype and the frequency of the recessive phenotype. while equation 2 will permit us to determine the frequency of the homozygous dominant genotype, the frequency of the homozygous recessive genotype, and the frequency of the heterozygous genotype. And now for Handy-Weisberg equilibrium to hold, for this equation to hold, okay, for it to hold, there are certain conditions that has to be respected. The first one is that the population should be large, okay. So this actually explains why Mendel's crosses with his pea plants were actually having changes in genetic frequency. The reason is because he had a small population, okay. So for this equation to hold, the population should be large, okay. The second condition is that mating should be random. For the population to be large is so that when there is a change in the frequency, it should not highly affect the population. When the population is small, the change in frequency highly affects the population, okay. Mating should be random. It should not be selective, okay. So you mate with anybody you meet, because if mating is selective, it means that some individuals will be favored, and when they are favored, they will have more mates, and when they have more mates, most of their genes will be propagated in the population, and that will lead to the change in the genetic frequency of that population or in the gene pool, okay. There should be no gene flow, okay. No new genes should be introduced into the population, because new genes are introduced into the population is going to change the genetic frequency. There should be no mutations. Of course, mutations will lead to the change in the genetic frequency, okay. There should be no migration, okay. People should not migrate into the gene pool or into the population, or migrate out of the population. If they do that, then there will be a change in the genetic frequency. So these are the conditions that need to hold in order for the Handy-Weisberg equilibrium or his equation or their equation, in order that this equation should be correct, okay. Now we move into speciation. What is speciation? Speciation is the process which leads to the formation of new species. What are therefore species? Species are organisms that can interbreed to produce fertile offspring. We should note that definition very clearly, okay. So species are organisms that can interbreed to produce fertile offspring. So if you have two organisms that interbreed and they do not produce offsprings, that fertile offsprings, they are not of the same species. If we take for example a horse and a donkey that resemble, okay, if a horse, since they resemble, someone can say that they are of the same species, okay. But if a horse should mate with a donkey and then they produce an offspring, and this offspring is not fertile to produce other offsprings, it means they are not of the same species, okay. And here under speciation, we have the concept of inbreeding and outbreeding. Inbreeding is actually the mating between closely related individuals. We have an example between parents and their offsprings, or brother and sister, while outbreeding is mating between unrelated individuals, unrelated or less closely related individuals, okay. Now, the concept of breeding, some people disagree with the extent to which the relationship is actually considered to be closely related, while some agree with this definition. So the extent of relationship, whether it is closely related or less closely related, is not really defined. There is not actually a barrier that actually define the degree of relationship between individuals. But this is the most appropriate that we can get to define inbreeding and outbreeding. Inbreeding, that is a breeding between individuals that are very closely related, and outbreeding is the breeding between individuals that are not related, okay. Now we have come back to this notion of genetic drift. These are chance or random events which tend to cause changes in the gene frequencies in the gene pool. Okay, we have two types of genetic drift. We have the bottleneck and the founder effect. Let's take the bottleneck effect. It is a situation where by a natural catastrophe randomly eliminates individuals in a population irrespective of their genotype, okay, while allowing some remnants behind. This also is very important, okay. The natural catastrophe should be random, eliminating individuals irrespective of their genotype, okay. It should not select certain individuals with particular genotypes. It should be random, okay, and it should eliminate individuals randomly, and it should also allow a remnant behind, because if you eliminate every individual in that population, it is no longer a bottleneck. So it is only a bottleneck when it eliminates people randomly, okay. It is a natural catastrophe that eliminates people randomly and allows a remnant behind, okay. That is known as a bottleneck effect. An example, we have earthquake. An earthquake can happen here and then it will kill some people irrespective of their genotypes and then allow some people, okay. And then we also have a disease like Ebola, okay. Ebola comes in, it eliminates people irrespective of the genotype that they have. So it attacks anybody, attacks anybody, and some will die and be eliminated from the population, while some will be left behind. So that is a bottleneck effect. Then a founder effect is the second type of genetic drift. It's a situation where a small group of individuals are separated from the rest of the population and then move into a new area. So that new area they move into, they have actually founders in that area. Okay, so that is the founder effect and it's a form of genetic drift. And we should note here that it should also be a chance event. Might be you have cattle that is moving on a grazing land, okay, and then an earthquake happens and divides it into two, such that a small population of that cattle moves into a new area and is separated from the other guys. So they move like that and they live separately and graze separately like that, okay. So that is some sort of a genetic drift, okay. So